Hello, everyone. Welcome to our fourth and final session of the year of the Signals Research Suite Quarterly Connect Community Series. We released Signals Research Suite last year as a complete unified SaaS software for scientific research, and in this fourth Signals Research Suite Quarterly Connect, we take a look at how to query your data for structure activity relationship analysis in Signals Research Suite. My name is Chris Stump, and I'm the product marketing manager for Signals Research Suite and Signal Synergy at Revity Signals, and I will be your moderator for this session. We have a fantastic webinar for you today, but before I introduce today's speaker, I want to share with you what I think that you'll learn today in today's webinar. So what we're going to focus on today is how you can use Signals Research Suite for self-guided data retrieval and structure activity relationship analysis. We want to help you identify the best experience for your searching. And I want to point out that we recently added the integrated search app to Signals Research Suite. And this can help provide you with a guided template-based search, or you can also use a quick query that kind of gives you your data very quickly if you just want to see your data now. I should also point out that you will hear Samantha speak about Inventa today during the presentation, and this application resides in this data-driven analytics area. So that's the Inventa enables the data-driven analytics capability, and this is at the decide stage of the make, test, decide within Signals Research Suite. Okay, so after Samantha speaks today, we're gonna have some time for some Q&A. So that'll give you an opportunity. If you're watching and you have a question, you can ask her at the end of her presentation. So now I'd like to introduce Samantha. It gives me great pleasure to introduce Samantha Jashonik. She is product manager for data-driven analytics within Signals Research Suite. She has a PhD in molecular biology, cell biology, and biochemistry from Brown University. Before Samantha starts us off with the presentation, I just want to give you one housekeeping reminder. This webinar is being recorded, and the recording will be shared with all attendees in the next couple of days. So watch your inboxes for that. So without further ado, let's jump into the presentation. Samantha, the floor is yours.
Thank you very much, Chris, and thank you everyone for your attendance today. As Chris mentioned, my name is Samantha Jashonik, and I'm the product manager for Inventa. Inventa is the decisioning analytics component of the Signals Research Suite. And I'm excited to talk to you about that today in the context of how to query your data and use that data in your SAR analysis. Now, one of the fundamental prerequisites to decisioning analytics is really the ability to retrieve a subset of your data. The notebook features of the Signals Research Suite are very open-ended in the types of ways that data can be captured. So we understand that scientists, whether they are in biotech or pharma or industrial chemistry or formulations, they really need the freedom to record their data in the way that's most meaningful for them, whether that is with free text or images or tables or worksheets. But at the same time, we also recognize that not all data is created equal. So, for instance, daily details about your cell health during passaging, they don't carry the same weight necessarily as a routinely performed assay that's now in the late stage of a screening campaign. And so the inventive features of Signals Research Suite are really designed specifically with your high value data in mind. Little fun fact for today is that the name Inventa actually comes from the Latin for discovery. And so within Inventa, we've built tools to help you discover that eureka moment, which should allow you to advance your material, your entity to the next stage of development. And data published from Inventa, it can come from lots of different places. So it could be from the material library, could be in a table and experiment, output of an in vitro workflow or an in vivo workflow from maybe flat files, or from a report from a CRO. But unlike those areas of signals that allow for that free-form data capture, by the time you are desiring your data to go into Inventa, we want to clean it up. We want to have a data standardization process. And so the way that we handle that inside Inventa is that we empower our data stewards to define their results schema. So they do this through an easy to use graphical user interface. And in Inventa lingo, what they are creating are measurement types. Each measurement type contains a set of attributes that describe it. And so by mapping the source data to the expected data schema, and again, this is done in a simple point and click method, we can ensure that the data published to Inventa is in the same structure and using the same consistent vocabulary. Consequently, Inventa is able to query your high value data extremely effectively. And we have a few different methods for data query and data retrieval in Inventa. Our traditional method has been around since the beginning. It's called the Global Search App, and it allows for really flexible, extremely granular, nested, and what can be complex queries. As we've gotten feedback from our users of the Signals research, we realized that there was really a desire for a different type of search experience though. One that provided maybe less flexibility, but instead opted for more templated search, more controls enforced by admins, and such was born the Integrated Summary app, which you may also hear me to refer to as the ISA. And so today, my goal is to demonstrate the Integrated Summary app and discuss how it can be used in the retrieval of data and the SAR analysis of that data. What I'd like to do is really begin at the end. I want you to envision how a user or you might engage with the Integrated Summary app, how you would use a fully pre-configured query template. We'll go through and we'll discuss potential flow of interacting with your data. I won't be able to help myself, so along the way I'll show off some neat Inventa and Signals Research Suite features, and then at the end we'll return to the beginning where we'll actually go in depth on the Integrated Summary app query tool and how you would configure it. To begin, I will log into my Signals Research Suite as the user Olivia. When I click this button, it's going to redirect me to my Signals dashboard. And right away, I notice that I have some notifications. I'm going to click on this little bell and it tells me that some templates have been shared with me. There's some comments on this templates. And so all I need to do is click the notification and that's going to take me directly to the object it's referencing. In this case, that's an Inventa analysis. And that Inventa analysis contains a pre-configured query for the Integrated Summary app. I also see within this a larger analysis object that a comment's been left for me that is telling me that the latest HTS data is in and I should go check it out. So I'm going to open this template. I'm going to do that by clicking on the View in Spotfire for Signals online. And note that when I click this, I get a notification that I only have read-only access. That means I can still interact with the template. I can still perform a query. I can click around in tables. I just can't overwrite the original template. And so that level of permission and control is one way that we empower team leads to set up these templates for, say, a larger group with the confidence that the work that one user like Olivia does won't change that original template for another user like Ella. So the first page that opens up here is the Integrated Summary App query template that's been predefined for me. You can see that my admin has already associated this with the correct data bucket or that's at the top of the screen. And they've also already selected the measurement type of interest. So even though this template is read-only, as I mentioned, I can still modify parts of the query. So maybe I want to further refine this very generic HTS search query, and I want to actually specify that it should be limited to compounds that are being tested in this protein phosphatase project. So I'll enter that here. And then I also want to limit it to the campaign stage. So I only want to return things that are in the primary screen. On the right, once I enter that, I can expand and actually see an exact preview of the data that I'm going to return. So because of the nature of this template, everything else about how that data should be aggregated and organized is set up for me, and I really just need to click run. If I didn't want to enter any specific query criteria, I could have clicked run from the beginning, but I did want to enter a few things, so I did that. And when I click run, the data is downloaded, and I'm given this query results tab. The query results tab reflects essentially the same thing that you saw on that results layout preview. So I see that I have both a summary table that contains material information like the structure and the molecular weight, as well as aggregated results data, which I can see has actually been organized based on the specific target being tested. So in this case, I actually have two different but related protein phosphatases, PP1 and PP5. And you'll note that nowhere when I was setting up that original query, even when I was modifying the conditions, did I have to specify that they were different phosphatases. The query and the way that it was set up was automatically designed to split that aggregation appropriately. So in addition to that, the summary table that we see up top here, I also returned the measurement tables that contains the individual results that make up the aggregated results above. And these tables are actually automatically linked so that clicking a row in the summary table automatically selects the related rows in the measurement table. Now, one nice feature of this app is that those tables that were downloaded as part of the query are actually downloaded into this dashboard, the part of the data canvas, and they can be used in other visualizations. So if I'm Olivia and I'm looking at this HTS data, I may decide that I really want to build out an analysis with some more visualizations, so maybe I can make some decisions on which compounds should be sent for secondary screening. I did tell you at the beginning that I can't overwrite this template, right? My admin has blocked that because they haven't given me write access on it. But what I can do is I can go ahead and save this template as a new Inventa analysis. And so here I'll go ahead and save it as Olivia's HTS analysis. I'll put it in the same protein phosphatase 5 inhibitors project notebook, and you'll see in a moment that this will update, and now I am inside sort of my saved version of this dashboard that I can freely modify. So then I'll continue on and I'll say create my visualizations. I personally like to set up different pages that are relevant to the particular type of analysis that I'm doing. So in this case, I've set up a tab. I've called it HTS analysis, and I've now leveraged those tables that were downloaded with the query. You can see that on this page I've added coloring to mark different hit criteria. I've created a scatter plot and also a Venn diagram, all looking at the performance of these compounds against those two particular targets. Now, in my case, I'm particularly interested in the protein phosphatase 5 target. The PP1 target I'm testing as a counter screen. I want to get a compound that's going to be very selective to my protein phosphatase 5. And looking at this page, I can see that over 300,000 of the compounds tested from sort of a general screening library, only a fraction of them qualify as hits. And so at this stage, what I need to do is let Ella know then which compounds to run through some confirmatory assays. So looking at this Venn diagram, again, this is all interlinked with the tables that have been downloaded. I see that there are some compounds tested that are not very selective. They are meeting the hit criteria for both of those targets. So I can go in and I can click and I can explore that space and see if there's anything interesting. And yep, there seems to be, maybe this is interesting, an oxybicyclo substructure. Maybe that's useful later when we're trying to optimize the structure for selectivity. So I'm actually going to go ahead and maybe send all 500 of these protein phosphatase 5 hits to Ella. One way I can do that easily is by selecting this section of the Venn diagram, and you'll notice that's highlighting the compounds on the left side of the screen. And then I can send that list directly to my Inventa analysis object so that Ella has it on hand. So I'm going to go ahead and do that, and then return to Signals so I can share this Inventa analysis with Ella. So I'll go in and I'll set the sharing, add Ella, and give her read access. All right, now imagine that some time has passes and Ella messages me, whether it's in the comments or elsewise, that my results are ready. And I can now reopen that same dashboard I was working in, Olivia's HTS analysis, and I'll slightly modify my query. So I'm going to change the stage here to also include confirmatory results, and then I'm going to press run. And I'll use the opportunity, since I'm in my own dashboard, to update some of the visualizations. So imagine that I've created another page here called confirmatory, and we're going to jump in and look at that one. So looking at this data now, I can see that most of my compounds tested in this confirmation screen were in fact selective for protein phosphatase 5. That's great. And now I need to choose a subset for some more intensive assays, like a concentration response curve. I've set up a distribution plot here, again, one of the features of the Signals Research Suite. I'll go ahead and highlight, and I want to pick sort of the top subset of data, which I can easily find in this violin plot. It's about 60-ish compounds. And once again, I'll save and I'll share that with Ella. I really like that when you save and you kind of export these tables to Signals, it automatically date timestamps it in the title. It gives a really good snapshot of the data. So as you could see from the two different times that we looked at this, both in the HTS the primary screen lens and the confirmatory lens, these dashboards are living and breathing. They're updated as new data is coming in. But there's certainly relevant times when you want to take a snapshot of the data. And so that's really what these exporting visualizations or tables or pages are for. And having that timestamp there is just a really nice convenience feature. All right, so some time has now passed, and Ella has done a number of additional assays with her team. She's been publishing those into Inventa, and maybe my team lead, knowing that we have a number of different assays coming in now that need to be analyzed, maybe they decide to set up a more of a late stage query template, and maybe they've shared that with me. So I can go to my project notebook here for my protein phosphatase 5 project. I see within it there's a new query template for late stage analysis. I'll go ahead and click this. Again, I only have read access on this one, but I can go ahead and open it up and see the query that my admin has set up for me. Just like with the earlier query, I can go ahead and I can set some criteria. So maybe I will come in and I will set a value for the aggregate IC50 results, saying that I only want those that are values returned that are less than 200. I can add some criteria on additional assays if I wanted. So the way that these assays are stacked, I'm requiring that a primary HTS screen has been run on the compound, and that any one of these other confirmatory or secondary assays has also been run. You'll notice that some of these I've given specific cutoffs as well. And one thing that you'll further note is that in both the criteria and measurement parameters section, the summarization is really able to be set on a per attribute level. So the percent inhibition, for instance, is going to return an average, but the IC50, which is more exponential, is going to be calculated with geometric mean. And we'll go through in a little bit how the admin has, actually limited the number of approved summarizations that can be performed on these different attributes. Another thing you'll notice on this page, now that we're dealing with more assays, is that the result layout preview is much more popular with data tables, so we can expect to get a lot more back. So now I'm going to go ahead and I'm going to download these datas. And just as I saw on that results layout page, there's a lot more going on, plenty more tables. And I can, of course, expand one of these tables to get a better view. I can click through the arrows on top to kind of scroll through the tables, but my personal preference when building a dashboard is to really set up pages that achieve specific goals or answering specific questions. So instead of doing everything on this single page, what I want to do is set up a new tab, and I want to really dive into maybe those IC50 curves. An important aspect of the integrated summary app is that even though we've simplified and we've flattened the query a bit, we really still maintain the idea of data relationships and drilling down. So you've already seen the data relationships in the way that the summary table and say the measurement table are linked together. And now we can emphasize that drill down as well. So when I did my query, it was set up that not only did I return a summary table and the individual IC50 results table, but also the corresponding well-level details to that IC50. And all of those can be graphed accordingly. So that's what you see at the bottom here. I've gone ahead and I've plotted those concentration versus percent activity. I can see multiple curves on the same plot. I can kind of configure this however I want. So that as I'm looking at this data and I'm thinking about, what should we do next? Maybe I want to get a better idea of the compound scaffolds that are performing well. Remember, I started with a pretty broad 300,000 compound library, and now I can, with these results, I can start to figure out modifications to make to those compounds to generate different leads. And so I'll start by just using one of the built-in inventive features, which is a scaffold extraction. And then I can drill down and find out which scaffolds are producing the most selective hits for my target of interest, which is that protein phosphatase 5. So you can see the graph at the bottom. I have the scaffolds along the x-axis and then number of hits on the y. So it becomes pretty clear that one of these scaffolds is performing better than the others. It happens to contain a benzamide. So from there, I can then perform, again using out-of-the-box functionality, a R group decomposition on that scaffold. There's not a lot of unique compounds with this particular scaffold, and maybe I want Inventa to help me determine what to synthesize next. So what I could do is use the bioisosteer replacement tool within Inventa, and that will allow me to think of additional compounds that I can make that vary in their R groups that are going to be more biologic active or stable. Or maybe, while I'm trying to figure out different compounds to synthesize, I also want to look in depth at a single one of the compounds and maybe investigate, what else has been done on that compound, perhaps in other projects, maybe in that deubiquitinase project that my colleague is working on. So I can go ahead and I can open up our quick query mode. And quick query, as I said, allows me to quickly bring back everything about a single compound. So in this case, with the quick query open, I'll go ahead and enter that compound ID, select it, I get a preview, and then I can select download, and in that single click, right, it's bringing back all the data that we have in the system about that particular compound. And at this point, right, the cycle continues. There's more making of compounds, there's more testing and deciding and just rerunning through that cycle. And eventually, if a compound is super promising, Signals Research Suite has apps to handle testing it in vivo with an in vivo study designer, the ability to look at PK parameters and calculate those, and even the integration of your in vitro efficacy data and that in vivo PK data all in one integrated graph to assist with candidate selection. But again, all of that late stage discovery had to really start with just first being able to find and retrieve your initial set of data. And so I hope in this first half, I've really given you a peek in how these new query tools can be used in combination with other great features of the Signals Research Suite for your SAR analysis. So in the second half of this Quarterly Connect, I really want to dive in more detail into this new integrated summary app and talk about how it's set up and configured. So let's go ahead and start with a brand new analysis and a brand new query. So again, I will go to my Signals Research Suite. I'll say create new Inventa analysis, tell it that immediately I want to preload the integrated summary app standard query, and I'll click go. When this opens, we have what's considered a blank query loaded. And if you look from a high level, there's a few main sections. So there's a material section here for really setting the criteria on your compounds or biosequences or other entities. There's a measurement section for selecting which measurements or assay results you want to return. And then there's a parameter section to further refine the subset of results from that data. The rest of the page has different summaries and preview layouts that update with the changes you make to the query itself. So one of the reasons that I've been referring to these queries as templates is because you're able to set up these blank query options, which allows the user to easily fill them in. So you'll note when I immediately open this brand new query, the chemical structure and molecular weight were preset attributes. On the batches, the batch project ID was already preset for me, and those are showing up because a data administrator defined it as such. So if I instead worked in a more biologics driven company, maybe instead of showing the chemical structure here, I would show a blast sequence search, which the ISA does support. Or if I worked in formulations, maybe as a data admin, I set this up to display components and their ratios by default. The point is that we wanted to allow admins to configure this tool so that whenever a new user came to it, there would be things that were preset for them. So it could be as simple as like the user comes in, enters a molecular weight or a substructure, and then clicks go, and everything else has been preset for them. Let me go and just fill this in a little bit with benzamide and molecular weight. And then point out that, I'm not just limited to the attributes that my administrator has, preset for me. They've done so to make it easy, but of course I am not limited to just the attributes that my admin has added. The templates and having those attributes preset is a way to make it easy to get started, but of course I can go in and further refine what I want added to this query. So I can add something like hydrogen bond donors just by this drop down, and I can do the same thing for my batches as well and add a property there. So there's a few things that I want to note as I've kind of added different fields to this query. That criteria summary is updated in real time. So I'll go ahead and add another attribute in it. As I do so, you see that attribute is being reflected inside the criteria summary, and any values that I specify are also reflected there. In addition, if you look at the results layout, that's updated too. And one thing to point out is, these sections are really what you see is what you get. So since I have 1, 2, 3, 4, 5 compound attributes listed, I see only those compound attributes listed in my summary table in that order. Of course, I can drag and drop them. I also see only that small subset of batch attributes I defined. If I added another attribute, right, it would be added to my criteria summary, and I'd also see this table update. So next in my measurement selection section, I need to choose which measurements or assays I want to explore. And remember, again, here the data steward ahead of time has already defined sort of my result schema or those measurement types that are available to me. They've even organized them with tags. And these tags make it so I can reduce or narrow down my list of measurement types by selecting combining tags. My list here isn't so big, right? It's just a few assays. But if you had, something more comprehensive, being able to narrow it down in this way is really convenient. So I'm going to select this tag, which is set for, the protein phosphatase project, and it's going to limit it to just my protein phosphatase assays. I can also select HTS. It's going to then add any assays that are for high throughput screening. And instead of maybe looking at, both of those tags separately, I want to look at the intersection of assays that fall under both of them, and so I can do that by just switching the tag setting up here. And of course, right, if you know your measurement type by name, you can also search for that directly instead of having to use the tags. So now I will go ahead and I'll select a particular measurement type, and you'll note that as I do so and I highlight this measurement type, the measurement parameter section on the right populates. And what's being shown here is really a subset of the attributes for this measurement type. So this measurement type actually has many more attributes. It could have dozens, and that can be kind of overwhelming. So one of the controls we give to the admin is to really specify the default visibility of certain attributes so that even when you make a new query, the most important attributes when you select any particular measurement type are immediately shown. And the idea is really like if you come to the query and you need to get a bunch of aggregate results for a few different measurement types, and you don't know, you don't really need to dive specifically into a super detailed query. You just add them to the query like I'll do here, and then click go or click run, and there's no other consideration really needed. So to further that point, I want us to hone in on the summarization method column in this parameter section. And as I mentioned earlier, the summarization has already been defined by the administrator. So we put the power to specify the appropriate ways to aggregate attributes in the hands of our data stewards, who should be subject matter experts here. So in this case, our data steward has set a different aggregation for IC50 and percent inhibition. So if I look at percent inhibition from this particular measurement type, I see that it's an average. But if I go ahead and I select the IC50 from this measurement type, I can see it set as geometric mean. And the admin, knowing that IC50 is more exponential, has set that to the appropriate method for analyzing it. But what if you want to support some additional common aggregations, maybe standard deviation or count, or just you want to concatenate the values? Well, those can also all be pre-approved by an admin, and the user can always enter this gear icon to select the attribute of interest and then add any additional of the pre-approved aggregation formats. So for IC50 here, you'll see that my admin also says that standard deviation, concatenation are also totally acceptable. So I'll go ahead and I'll add those. And then while I'm at it, I'll also decide to add another attribute like hill slope and substrate to my report. Doing so, let's take a glance over on the right side again at our report layout and look at what's been added. So for the additional summarizations on IC50, we see those all laid out in the table in the kind of the order that they were added here. We also see some additional attributes all together, like the hill slope. And if we added more measurement types, we would see those added to the aggregation at the end as well. The additional summarizations, I think are a really good illustration that this layout is very much what you see is what you get. You're seeing if you have a summary method in your parameter section, it's going to show up in your table. Now, let's look back at the measurement type and talk about the two other features we see here. So one is a checkbox to easily bring back any available well-level data. So remember in the first half of this demo, we were looking at those concentration response curves. Well, those were generated based on the well level details, which are associated with this parent assay. The other checkbox we see here is to split summarization by experimental factors. And so experimental factors are a way for you to get more reusability out of a measurement type. So for example, you may perform an assay that's done essentially the same way, but there's a variable like a target or a cell line, and these slight variations shouldn't dictate that you create a whole new measurement type. You should be able to just set it as a variable. And that's really what experimental factors are, and they are predefined by your data steward. But if there are any experimental factors that have been predefined for you, right, you'll see the user will see this checkbox and they'll be able to select it. And then selecting it ensures that your data is split appropriately. So if you remember back to our early demo, we had 2 protein phosphatase results, one for protein phosphatase 1 and one for protein phosphatase 5, and that data was automatically split and aggregated appropriately because we had used this experimental factors checkbox. The other thing I want to emphasize is the way in which this query is being constructed. So the layout of our columns here is extremely intentional. So I told you in the beginning, the integrated summary app approach takes a less flexible query approach in favor of more admin guardrails. And so how we've achieved that is really by flattening the data, which means you are either querying summarized data or unsummarized data, but you can't do both simultaneously. So if you want to do that, you're perfectly welcome to, and the global search app would be a perfect solution for you. So in this example, I am running a query where I want to find results where the geometric mean of the IC50 is less than 10. That's the exact order that these columns are in, and that's reflected in the summary panel as well. The summary table is only going to have rows with compounds that met our material criteria that also have a geometric mean of IC50 less than 10. However, we've also made a point to be really transparent with your data, which means that despite limiting the summarization value to less than 10, we don't actually want to limit the input data or obfuscate the data that went into that summarization value. So you may have measurement results that are 8 and 9 and 8.5 and 11, and all of those in this scenario would show in your measurement table. So let me go ahead and give you a more concrete example. So for this example, I am going to configure my view so that experimental factors are split by rows instead of columns. I'm going to set 2 conditions. So I'll set the target to equal protein phosphatase 5. And then I'll also set the average percent inhibition to be greater than 50. Let me go ahead as well and draw on this structure, which is the interesting oxy bicyclo that we saw earlier. And I will also enter a pretty narrow range for this molecular weight. Click Run, and you'll see that all the rows in the summary table have a value of less than 50. But when I look at the individual results, all 4 values that make it up are shown. So there's both those over 50 and under 50. And so that's what I mean by full data transparency. We also do understand, though, that there are folks and there are personas that want to really filter directly on that measurement table. Really good, again, for scientists who are actually running the assay, who just want to look directly at the results. Useful for companies that sort of pre-summarize or pre-aggregate their data in another system and want to load those into the Inventa aspect of Signals Research Suites. But for that scenario, we allow you to run an unsummarized query. So I would go ahead and return to that gear icon, set it to be unsummarized, and now you see that summarization method column has been removed. And so now I'm querying directly on the attributes themselves. So I'll go ahead and again I'll set the IC50 is greater than 50 here, and the other conditions I specified previously. And then when I do so, you'll note that all the rows that are returned in this measurement table, they are all over 50. You also have a kind of message at the top alerting you that this data has been filtered, or it's has some conditions set on it. We wanted to introduce that because we know how often that data gets either exported, whether into Inventa Analysis or as a visual, or put into a PowerPoint or a PDF. And we felt it was important to at least provide that indicator that there's something, that this isn't the full set of your data that's being represented. As you can see, there's still flexibility in the integrated summary app that allows you to define the way you're going to query your data and retrieve what you need for SAR analysis. Our goal with this app was really to create something that provided more admin regulation, requires a bit more admin setup at the beginning, but then allows team leads to create these query templates that are just really easy to adjust by the end users. But we had also continued to hear from customers that they still just wanted a quick way to retrieve all the information on a particular thing in their system, and so that's why we also included a quick query mode. And the quick query mode is the scientist's answer to give me all my data now, right? So I'm going to go ahead and open the quick query. And here you can, very few inputs, you can enter an ID, you can draw a structure, enter a sequence, and then you'll get a list of matching entities. I'll select one, and you see I'm given an immediate preview of their attributes. And from there, I just click run, and I return all the data about that compound. If you wanted to, you could modify the quick query by limiting it to a certain date range, or splitting it by experimental factors. And then if you want to go further, right, you could, instead of focusing this on a particular entity, actually focus on a particular assay or particular measurement. So here we're going to select this rat assay, and it gives a summary of the assay with some description and its tags, and then we can click run and see all the tables that are related to it. This has gotten some interesting use cases where, say I want to keep track of a particular assay and I want to create a template for the latest week's results. I can actually set the date condition here to this week and save that as a separate Inventa Analysis. And then this week remains relative to the time of opening that analysis. So whenever you open this analysis, you're getting the latest and ideally greatest information for this week. So we've chatted for about an hour now, and we've only really scratched the surface of the full capabilities of the Signals Research Suite. I hope that I've demonstrated today how you can use the power of the Signals Research Suite in self-guided data retrieval and SAR analysis. If you want to recap, the Research Suite empowers you and your organization to decide the best search experience for you and your users. So if you want ultimate flexibility and configurability, we recommend the Global Search app. If you want more guided templates with guardrails from admins, check out the Integrated Summary app. And then, of course, I just think anybody who's using the system can benefit from a quick query to get me my data now. And so with that, Chris, maybe I pass it back to you and we can take any questions.